Animals do not forage randomly. Their food-seeking behaviors follow the Earth's 24-hour cycle. For example, many wild animals search for food in the morning and evening, while avoiding the midday heat and predators. Interestingly, this rhythmic pattern persists even in laboratory conditions, highlighting the critical role of the internal circadian clock. In animals, central clock neurons regulate circadian rhythms. But how do central clock neurons connect to neural circuits that drive rhythmic behaviors, such as foraging? Olfaction plays a key role in foraging, providing information about food sources, including their type, amount, distance, and quality. Importantly, olfactory sensitivity is not constant. It changes with environmental conditions, internal states, and even time of day. This raised an intriguing question. Could circadian modulation of olfactory circuits underlie rhythmic foraging behavior? To study circadian foraging behavior, we placed flies in an arena where apple cider vinegar was applied at the center, and monitored their foraging for 10 minutes. We observed that wild-type flies display a clear foraging rhythm, with 2 peaks in the morning and evening, and 2 troughs at midday and midnight. In our study, we discovered that one group of GABAergic inhibitory olfactory projection neurons, called MLPN3, shows circadian responses to food odor and plays an essential role in rhythmic foraging. Notably, blocking GABA release from MLPN3 neurons induces an additional foraging peak at midday. Moreover, whole-cell patch clamp recordings revealed that MLPN3 neurons exhibit rhythmic firing activity, with lower spike frequency and average membrane potential in the morning compared to midday. These results indicate that MLPN3 neuronal activity is lower in the morning and higher at midday. And this kind of rhythmic pattern is driven by dopaminergic inhibition from a specific group of neurons. Furthermore, these dopaminergic neurons are activated in the early morning by circadian morning cells through the neuropeptide PDF. Optoinhibition of morning cells significantly disrupts morning foraging. Remarkably, this impairment can be fully rescued by inhibiting MLPN3 neurons. Together, our findings reveal that rhythmic foraging is orchestrated by a disinhibition circuit. The circadian clock activates dopaminergic neurons in the morning, which suppress the inhibitory MLPN3 neurons, thereby lifting the suppression on foraging. This study uncovers a fundamental mechanism linking the circadian clock to sensory processing and behavior, highlighting the role of dopamine-mediated disinhibition in circadian regulation. Read the full paper in Current Biology.